Hello and welcome to today's webinar on garden weevils. My name is Richard Fennessy and I'm a grape and wine research scientist with the Department of Primary Industries and Regional Development, also known as DPIRD. I start by noting that DPIRD acknowledges the traditional custodians of country, the Aboriginal people of the many lands that we work on and their language groups throughout Western Australia, and recognise their continuing connection to the land and waters. DPIRD respects the continuing culture of Aboriginal people and the contribution they make to the life of our regions, and we pay our respects to elders, past, present and emerging. In today's webinar, we'll be hearing from 4 speakers covering a range of previous research on management techniques of garden weevils in Australia, new research out of South Africa on the effectiveness of biological control agents, and insights into recent trials in Margaret River. Should you have any questions for the speakers, we'll be having a Q&A session once the presentations are concluded. Using the Q&A function on the top right of your screen, please write your question and note the speaker you would like it addressed to. I will now introduce our first speaker, Alison Matthews, who is a research scientist with the Insect and Disease team at DPIRD based in Manjimup. Her work focuses on sustainable pest management across a wide range of industries in the southwest of the state, including pome fruit, stone fruit, olives, vegetables and grapes, with her current projects focusing on avocados and truffles. Alison will be reviewing the major garden weevil research that has taken place in Australia over the last few decades. Much of this research was led by Stuart Learmonth, Alison's predecessor at DPIRD, whom Alison worked alongside for several years before his retirement. I'd like to now welcome Alison.
Thanks very much, Richard, and hello everyone from, I think we've got Australia, South Africa and New Zealand people in attendance today. I'm going to be turning my camera off for my presentation just because my internet connection isn't the greatest at the moment, but I'll still be here. Okay, so as Richard said, I'm going to be going over some of the past research that's been done in Western Australia on garden weevil, just to give a bit of a recap of what's been done and where we're at here. So these are all of the projects that have been done based on garden weevil over the last sort of 20 to 30 years. So the first couple that I've got listed there were focusing on garden weevil in pome fruit, but some of the findings from those projects are still relevant to viticulture. And so as you can see, the last, I guess, official project was back in 2011. But Stuart was definitely still playing around with weevils right up to when he retired just a few years ago, because it was, I guess, their control was one of those problems that he was never able to crack in his career, and it certainly gave him a lot of challenges. So I'll just be running through these projects chronologically. So this project here was one, like I said, that was based in pome fruit, but had relevance to viticulture. So it really focused on the orchard floor species and then also nematodes for management. I've put up in there in the dot points some of the major, I guess, findings from that project. So they found that clover alone led to lower survival rates of garden weevil larvae, but when it was in combination with apple trees, they fed on those preferentially, so the survival wasn't reduced at all. And it turned out it was actually the grasses like ryegrass, kike and oats that resulted in the shortest survival times and the lowest egg numbers for the garden weevil. And the project did conclude that using parasitic nematodes was not feasible in orchard conditions. So they worked well in the lab in bioassays, but the orchard conditions at the time of the releases in spring and autumn just weren't ideal for them to work. And that was put down to low soil temperatures and low soil moisture at the time. So this next project was led out of Victoria and again focused on pome fruit, so mainly apples. but there were some things relevant to garden weevil that I've listed there. So they tried the sticky band technique as well, that I'm sure you're all familiar with. And they slowed access of the weevils to the canopy, but didn't prevent it. They were trialling some different attractants to use with baits, with chemical baits for attracting and killing the weevils, and found that carrot and apple worked quite well. There were bioassays again of nematodes. So the species there, Zelandica, which is now commercially available, and that was very promising at the time of this research. They did a survey out in the field to see if they could find egg parasites, like naturally occurring ones that could potentially be used for biocontrol, but unfortunately they didn't find any. And then there was also some work done on poultry, and that was shown to have some success. So this next project, which concluded in 2013, was largely focused on insecticide and trying to find different insecticides compared to the ones that were registered at the time. I'm not really going to go into the detail of that because I guess we're not really focused on insecticide use in these presentations today, but just, yes, making you aware that that project took place and that research occurred around that time. This next little bit of research in 2005 was a short series of lab trials done by a student, and again looking at trunk bands, and there were different types of exclusion bands that they'd tried, and they found that the glue one on the left, the sticky barrier glue, and also the skirt style of band, which is the one on the far right, were the ones that were most effective. There was a little bit more work done on exclusion bans in this larger project that Stuart was involved with, finishing up in 2011. So they found that the exclusion bans, their use was a little bit inconclusive. In their trial sites, they didn't have as many garden weevil as they would have liked, so it was hard to make, I guess, conclusive results from that. And in the lab studies, there was, I guess, quite limited effect with the garden weevil fairly easily being able to navigate over the bands, and these were the Dacron fibre bands used in this one. But yet there were some vignerons that reported success in the field with using them. This project also focused a lot on having a weevil, I guess, a weevil watch, or garden weevil watch they called it, and it had weekly reports from vignerons across the region, and that was all collated and placed on the department website so people could see what was happening where and where numbers were increasing, which was interesting. But then the variability in weevils between vineyards, even within the same region, was quite significant. And so even though it sort of gave a slight indication of what was happening where, people couldn't apply that information to their own vineyards without having their own monitoring. So it was very much reliant on people having to do their own monitoring because the variability was so great. And there was another project that also wrapped up in 2011. This one had quite a number of objectives that it was trying to meet, and so there were 6 different objectives in the project, and I've just listed there the major outcomes from each one of those objectives. So the first one was about developing a step-by-step guide for growers on how to, I guess, methodically assess a new control strategy that they might want to apply in their own vineyard. So that was done and then made available on the DAFWA website. The second objective was related to developing a framework of extending information on garden weevils to industry to ensure that all information was getting out in a timely manner and in a way that people could access. So the Garden Weevil Watch was developed and reporting on results out of the other project, and then it was all the garden weevil information was being, I guess, more loaded onto the department website for people to be able to easily access. In the third objective, it was about testing, developing and testing the economic action thresholds for garden weevil infestation. So it was a way of, just determining how many you need to have before you're actually at a level where it's doing economic impact. That was found that had to be based on a foliage scoring, damage scoring system rather than the monitoring bands, because the monitoring bands, there wasn't a good relationship between numbers that were being found there and the actual level of damage being observed. But there was still, I guess, no really clear relationships between the damage being seen and the scoring and the level of weevil seen. So there still isn't a, I guess, a precise level of what is the economic impact and the threshold number for weevils. There was another objective looking at the garden weevil distribution within vineyards based on what's in the environment in that area. So, and the results from that one showed that there was no correlation between what was growing on the orchard floor and the weevil numbers. So differences in orchard floor species across vineyards and numbers of weevils related to that. There was a slight relationship with soil type and weevil numbers, but this was a bit skewed given the low range of soil types that were able to be assessed as part of the project. There was, again, looking at physical exclusion barriers. And this was not just the trunk bands that we've talked about before, but also the use of kale and clay as either a butt drench or a foliar spray as an alternative to chemical sprays. So the sticky barriers were effective, the fibre bands were inconsistent, although their effectiveness improved with the addition of chili on them, and the kale and clay was also inconsistent in being able to reduce damage. And then the last objective of this was to come up with an IPM package for garden weevil management for vineyards. So a lot of this information was then collated and put into that. There was I guess one interesting point in the discussion section of this report was that there was often a disconnect between the differences in weevil numbers between the different treatments and then differences in the yield in those areas. So it was difficult to determine exactly how effective a treatment was or not. And then also the question raised of then if in what cases was it actually a pest or was it just present there but not causing damage that was economically significant. And my last slide, I just wanted to put this up. So this came out of a project in 2015 that was pretty much trying to determine research priorities for the industry at the time in regards to garden weevil. There were quite a number of topics that were listed in the report. I've just listed a few here that came up as high or medium priority at the time. But I've just wanted to put this here for people to have a scan through, look to see what the priorities were there at the time, think about what I've said about these past projects that have happened in WA, but then also seeing what's been happening in South Africa as well. And yeah, just having a think about where priorities are now for the Western Australian industry, what potential areas there might be to look into the future for management options as well. So it's just a little bit of a seed to get some thoughts and some conversations happening. And that's...
Alison, sorry, Alison, can you just make sure that slide's come up? Hasn't come up on my screen yet.
Sorry, I clicked. Yeah, I'll leave it there for a while there for people to have a look at. So yeah, I mean, this was back in 2005, so it's quite a while ago. There's been some new knowledge developed since this review. Some new products have become available, some different entomopathogenic nematodes, for example, are now commercially available. So yeah, there could be things on the horizon for us to look at here in WA.
Great. Well, as everyone quickly sort of scans through that slide, just wanted to thank Alison for sharing a snapshot on what's been conducted predominantly here in Western Australia in terms of R&D and garden weevil, as we acknowledge that it is a significant pest for wine producers in the southwest of the state. So thank you, Alison.
No worries, thank you.
Worth noting at this point, and Alison briefly touched on it, is that we have been chatting to some New Zealand researchers about garden weevils, and yeah, hello to those joining us from New Zealand today. Garden weevil is starting to show damage across 3 New Zealand wine regions, and local researchers have conducted a preliminary 1-year study to identify the weevil species, the economic impact of the damage, and a review of control strategies. But an important observation worth noting today from this work was the incidence of garden weevils being transported across vineyards by machinery. I thought it's really worth highlighting this observation because it sort of reiterates the importance of controlling movement from known infected areas to clean areas within vineyards. Really appreciate the researchers in New Zealand sharing their findings with us, and we look forward to following their journey towards testing different spray options and investigating potential non-spray options in the near future. So moving on, I'd like to now introduce our next speakers. The first being Stefan Hansen. Stefan is a South African-based entomologist and researcher currently completing his PhD at Stellenbosch University in partnership with the Centre for Biology and Management of Populations in Montpellier, France. Stefan also has a Bachelor of Science in Agriculture with a double major in entomology and plant pathology, and is very passionate about supporting producers with information derived from research that contributes to effective management practices. Presenting with Stefan today is fellow researcher at Stellenbosch University, Murray Dunn. Murray completed his undergraduate degree in Bachelor of Science in Ecology in 2017 and went on to do a master's degree in nematology, which he completed in 2019. He's now in his 3rd year of his PhD, also in nematology. All his research at both master's and PhD level have focused on entomopathogenic nematodes, or EPNs, and specifically the artificial mass production of EPNs for use of biocontrol agents for important insect pests. So I'll hand it over to Stefan to start his presentation. Thank you.
Hi, sorry, still uploading slowly but surely, but I'm sure we're going to get there soon enough. Thanks for the interesting presentation, Alison. It's actually fascinating to note the parallels of what, and I'm sure many of the growers and people in industry in South Africa would have noted the parallels between what we struggle with here and what you are struggling with in Western Australia, and that these guys always have this sort of variable results. There's not seemingly any straightforward answer with garden weevil. Okay, we're about a third through with the download. Alison, while we wait, you mentioned that there's a Heterorhabditis zealandica commercial product already available in Western Australia that worked well in the lab but didn't work that well in the field.
Yes, yeah, that's right. So that was the one that was trialled quite some time ago. But there is another species. Did I mention that at the end there? I can't remember. You're going to have to forgive me if I pronounce it wrong. Bacteria. What's that one called?
Bacteriophora.
Yes, that's it.
Okay.
So that one is commercially available now, which I'm not sure if it was when these trials were done. So that's, I guess, a potential for one for us to be trialling over here, chatting to the suppliers of this product. Apparently it does work at slightly cooler temperatures. So that would definitely be an advantage for when we need to, you know, be putting it out in the field. So there are possibilities.
Wonderful. And has it been used on, or has it been trialed on garden weevil in the lab?
Not in Australia, no. I don't think so.
Okay.
And the suppliers weren't aware of anyone having trialled it in garden weevil, you know, having accessed it from them. So yeah.
Okay. No, cool.
Sure
I'll tangent off that in my presentation. Can everybody see the presentation?
Yep. Thank you.
Yep.
Okay. And the laser pointer?
All clear.
Fantastic. Okay, let's get going. Okay, so Richard basically already introduced us, so I think we can actually skip this slide. Okay, and jump right in. So what is garden weevil? Now, garden weevils belong to a subfamily of true weevils known as Intiminae, or the broad-nosed weevils. And what's significant about this is that many members of this family have quite a broad host range. So they've got the ability to shift from native hosts onto agricultural host crops and become quite important pests. Now, this particular species or species complex is native to the Mediterranean climate region of South Africa, so the Southwestern Cape. From here, it has spread to other countries of similar latitude, including Australasia. In its native habitat, it feeds on daisy family plants in the sort of habitat that you can see on the top right of the screen. And what complicates matters with this pest is that it consists of a complex of several highly similar cryptic species. However, based on records that have been sent to us from Western Australia and other older museum records, the species that we are dealing with in Western Australia and most of the agriculture in South Africa is Flectinus calosus sensu strictu, the true banded fruit weevil. Okay, so the South African perspective, here it is known as the banded fruit or the vine or the V-band weevil due to this band of white scales at the back of the wing casing. They're a multi-million South African rand problem on apples and grapevines and to a smaller extent pears, blueberries, plums and nectarines. That is not just damage but also the cost of control. The damage is both through primary damage, as you can see on these apples, and phytosanitary restrictions through hiding in these apples and hiding in grape bunches. There are concerns that they will get exported, so they are a pest of phytosanitary concern for our exporting countries. Okay, so how do we control this? And I just want to thank Dr. Eleonora Alsop, who's not here today, for providing this overview. We monitor them in orchards using cardboard stem bands. So it's a corrugated cardboard band that goes around the main trunk of your tree or your vine, and the weevils go and hide under there. And by monitoring this, you can sort of get an idea of what the populations are doing in the orchard and when to spray. Also on the feeding damage on water shoots of vines and trees. Those non-toxic sticky stem bands that Alison talked about are also used in South Africa, and that's sort of our main control strategy for these weevils. and also sprays, both of the foliage and of the raised beds, using broad spectrum insecticides like several pyrethroids, bioneem, and then some commercial entomopathogenic fungi in the genera Metarhizium and Beauveria. We are going to touch a bit more on that shortly. They are difficult to control due to the cryptic habits of the adults, the fact that the adults are very hardy. They're built like little miniature tanks, so they're not that easy to kill. The larvae and the pupae live under the soil level, and in a perennial system, this is quite difficult to get rid of, especially if you going organic and you can't use soil insecticide drenches. As with most insect pests, you do get resistance to chemicals over time, or at least there's been anecdotal evidence of that in Weevil, and also increasing restrictions on the chemical actives that may be used in South Africa for treating this and other insects. So the alternative to this is then biological control. There's been immense progress in South African entomopathogenic nematology, largely thanks to this lady, Prof. Antoinette Malan, who's both Murray and my co-supervisor. She's been involved with species discovery and descriptions, describing the symbiotic bacteria that are associated with those nematodes. Murray will touch a bit more on that shortly. Screening these nematodes against many of our key pests, including garden weevil, and then mass culture, in vitro culture of select nematode species. There's also been increasing exploration into the isolation and use and mass culture of entomopathogenic fungi in South Africa, also applying that on pests like garden weevil. Okay, so what are entomopathogenic fungi? They are soil-living fungi found worldwide. The infective propagule are Conidia. Here we have a microscope photo of a little conidium forming on a Beauveria fungal thread. So once this attaches to the insect, it grows into the insect and infects them, and those fungal threads start growing throughout that insect's hemicell, or its internal body cavity. It kills the insect in about 3 to 14 days through a combination of toxins, mechanical damage, and nutrient host depletion, or host nutrient depletion. And after the insect has died, a new spore starts forming on the outside of the dead insect on the cadaver. Now, as you can see on this weevil here and this weevil here, and those spores are then ready to go into the environment and infect a new host. So the main interest in biological control has been on the Metarhizium anisopliae complex, which are green spores, the Beauveria bassiana complex, which are white spores. The reason I put the species in inverted commas is that we also have species complexes in these fungi. So there are several species that are lumped under the anisopliae concept and that are lumped under the bassiana concept. And it is important to know what you are working with, as different species will have slightly different characteristics. And even within those species, you get strains. The best analogy I can come up with are, for example, different strains of flu, different strains of COVID that have different properties and different virulences. So the same thing happens with fungi. So obviously it gets commercialized under a sort of simple lumped classification, but on a research perspective, it is important to know what strain, what species you are working with, because they will have different properties, and it's not necessarily able to transfer information from one and extrapolate that to another strain. Okay, so here's another picture of Metarhizium and Beauveria on growing in culture. Here's the spores of Metarhizium and the spores of Beauveria. Okay, and then over to you, Murray.
Cool. Can you hear me, Stefan?
I can hear you, Murray.
Perfect. Cool. Thank you so much, everyone, for being here and listening to our presentation. As Richard so kindly introduced me, I am Murray Dunn. I'm from Stellenbosch University in South Africa, and all my research focuses on these wonderful little microorganisms called nematodes. So the nematodes I particularly focus on are entomopathogenic nematodes. Now, within the world of nematology, there are bad nematodes, there are good nematodes, there's some nematodes that don't really do much, they just do this in the soil and they're doing their own thing. But the ones that most people are aware of, the plant parasitic nematodes, obviously, because they parasitize our plants and they are really destructive to a lot of crop production all over the world. But these nematodes, these are the ones that work for us, the entomopathogenic or the insect killing nematodes. So when we've got insect pests in our orchards or on our agricultural land, and we've got insect pests, these entomopathogenic nematodes, or we just call them EPNs, they are a natural enemy of many of those insect pests. So they fit very well into a biological control as well as an IPM initiative. So as I said, they are natural enemies of many soil-dwelling insect pests. They belong, the two EPNs that we focus on the most are from the genera of Steinonema and Heteroditis. I'll get a little bit into that just now. And what's really cool about nematodes is that they've got this almost this weapon where they have a very specific symbiotic relationship with a bacteria of either genera Xenorhabdus or Photorhabdus. Now, it's not actually the nematode that kills the insect pest. It's actually the bacteria, and that's why it's its little weapon. So the way it's described quite nicely is their life cycle is that in a lot of the so-called popular science, they refer to almost as little farmers. And the reason is because they hold that bacteria in a special pouch inside their body cavity. And when they come across an insect or a larvae in the soil, you could almost look at it as a farmer approaching an arable piece of land that he wants to farm. So the nematode sees this piece of land and it deposits that bacteria, or as a farmer would, deposits the seed in the ground and waits for it to grow. Now, for a farmer, you wait for the plant to grow to a certain length before you harvest. The nematode does a very similar thing. So it deposits that bacteria inside the insect host, which happens to kill the insect, which is what you want. It waits for that bacteria to grow, and then it begins feeding on its so-called crop that it is now cultured or it's grown in this piece of land or the insect. And when there's no more food available, it then goes and seeks a new host. So in a way, they're just like these micro farmers that are going around looking for pieces of land to cultivate and grow food so that they can carry on living. So that's the basic life cycle. And once they've depleted that food supply, they'll go out and search for new insect hosts to reparasitize and start the cycle all over again. You can click next, Stefan. So if we want to use these nematodes in agricultural production systems, we obviously need a lot of them. And that requires mass production, either through an in vivo process, in vivo meaning using live organisms or an insect host itself. Or if we want to mass produce at scale, we have to go the in vitro route, which is artificial. Now, there are multiple companies around the world that are doing this kind of nematode mass production to give to a large, or provide enough nematodes to a large number of farmers all over the world. The main producers are these three companies at the top, Enema, BASF, and Coppert. I'm sure a lot of you are quite familiar with these three companies. As far as we are aware, and this was from a lot of the conversations we had at a recent conference in France that I attended, that Enema, BASF, and Coppert are the only companies in the world that are producing and formulating at a large scale. All other companies are, if they are mass producing, they're doing it at a very small scale in cottage industries. So the reason behind this is that it's very difficult to mass produce nematodes at that scale. And I'm talking about scales of 100,000 liter bioreactors and above. So very few people have the technology or have developed the technology to be able to do that. And that's something that we're also working on in South Africa, and part of our team is to begin that process. And I will explain a little bit more about how you do that now. You can go next, Steph. So mass production artificially, or this in vitro, takes place in either shake flasks, as you can see in the top left-hand corner and the right-hand corner, in these little shake flasks, and you place them on shakers, which provides oxygen just by that agitation. But those flasks aren't enough to provide to multiple farmers on many, many hectares of land. The only way to produce enough nematodes for that kind of scale is to mass produce them in bioreactors. As you can see in the right-hand picture here, that's a small bioreactor of about 500 liters at eNemo, the leaders in EPN production technology in the world. And on the left is one of their new facilities that I think it's been up for about almost 5 years now or so, that they've been producing nematodes in these 100,000 plus to 500,000 liter bioreactor containers. And that is enough to provide to a massive amount of farmers. And it's, I would say that this kind of situation is very difficult to get to because of the enormous R&D cost it takes to get there. So what you'll see in the next couple of slides is how we are trying to simulate or emulate what they're trying to do, but at a little bit of a smaller scale. You can go next, Steph. So in terms of the mass production globally, we now have to see, okay, so what is everyone else doing to compare to see if what we're doing is enough? Can we do this at a scale to produce enough nematodes for South African clients or South African farmers? And then what we would like to be able to do is then give off that information to other people around the world and teach them how to mass produce these EPNs at yields that are high enough to provide for your farmers. And the methods that we have developed have been able to do just that. So you can see the first Heterobdytis indica in red. That is currently the record for the number of nematodes, what we call an infective juvenile, the infective stage of the nematode. That is the record for the highest number of nematodes produced, and that is by Rolf Udo Ellis, who is one of the directors of that company, eNemo. So we've got a benchmark of how many we need to produce. And at the bottom, the two nematodes that are highlighted, or the one nematode with the two highlights, is Cynonema yerkelemensi. That is what the species that we are focusing on. Now, the first study we did, this was before I joined the lab. This was the lady that taught me all the work I know. She was able to achieve 75,000 IJs per mole. Now, compare that to what Ellis et al. Produced. You can see there's a vast discrepancy. Now, another research group studied the same species, and they managed to get yields of close to 300,000. In my recent work in my PhD, we have now gone above 300,000, and we range between 300 to 400,000 IJs per mole. So we're making progress in terms of the yield of production, which makes it more cost effective to get this product to the market. So we're making very good progress in being able to compete with those products that are already there, because you will be doing local production instead of importing products from overseas. So that is what's very key to this whole thing, is doing local production to keep down the costs. But to do that requires all of this very intense scientific research to pick up your yields and drive down the cost of producing this organism. Thanks, Steph. Next one. So the species that we are focusing on is this lovely little guy called Cynonema yerkelemensi. It was actually first isolated in Jerusalem in Ethiopia, and then later we found it in South Africa in a little province called Mpumalanga. So this is quite a small nematode, and that is very crucial. So the larger nematodes, which are about 1 millimeter in length, they struggle with micro insects, something like a fungus gnat, and they can't parasitize those smaller insects. So when you are looking to mass produce these nematodes, it's better to focus on the smaller ones because they have a wider host range. And this EPN fits into that category. And it is also a very good candidate to parasitize a wide host range, just based on the fact that it is a good candidate compared to species of the same size, or not species, sorry, of other species of similar size. So when you're isolating different EPN species, like my supervisor did, you then have to take all those isolates and all the different species and now do bioassays to figure out, okay, which is the nematode that we're going to look at commercializing based on a whole cohort of variables. Usually we focus primarily on, are they able to parasitize our economically important pests? And we slowly build up a database to see, okay, which one is our best candidate? And all the work that was done eventually pointed in this direction, that this was our best candidate across the board. It's a highly pathogenic species to multiple insects, but what is very key to their success is that they can be mass produced quite easily, and that has been the focus of my PhD. Next one, please, Steph. So what we've been able to do is mass produce these nematodes in these flasks at a very high level comparable to what is available commercially. Now, as I said at the beginning, this flask level production is not enough to provide nematodes to a large number of farmers on multiple hectares. But what we are doing is refining the process within these flasks to make the scale-up process a lot easier. And you can see in this photo on the left-hand side just how many nematodes are in here. So per flask, there's between 6 and 10 million nematodes, sorry, 6 and 9 million nematodes per flask, which eventually works out to that you need about 4 liters of media with the same yields to get enough nematodes for 1 hectare of land. So you can start seeing that the scales are quite large. So if you, for only 25 hectares of land, you need 100 liters of this media. So you can see very quickly how you need quite a lot of bioreactor or fermentation volume to be able to provide enough nematodes for a large agricultural land or farmers. But we are well on the way, and you can see that we have successfully produced these nematodes in this flask, and we are still in the process of optimizing that whole protocol to bring down the cost even further. Thanks, Steph. And the next step after the flask production was getting to the bioreactors. Now, this is what I was talking about earlier when I say we don't think we will ever get to the scale of e-NEMA, but we don't think that is necessary. Our goal is to create cottage industries in South Africa, and it might be something that the Australians can look at as well, is where you have very small local production teams that where you're able to produce your nematodes in these very simple bioreactors. Now, if you go online and you start doing research on bioreactor technology, you're very quickly going to get, a lot of it will go over your head, and you're going to get a bit of a fright at the level of technology that is required to do this. That's what it seems like, but we managed to do this with very, very simple bioreactors. I have no engineering experience, and we were able to, chatting to a glass manufacturer, we were able to design this bioreactor with very little knowledge to go on. And it had none of the bells and whistles that you will see on all these very fancy bioreactors, and we were still able to do it. So what it said to us is that the bioreactor technology doesn't actually have to be that complex. You just need to figure out a standard protocol of how to do it, and then you're able to produce enough nematodes for quite a large number of customers or farmers. So we've proven that this bioreactor technology isn't actually as complicated as it needs to be. Although I say that with a bit of an asterisk next to it, because we still do need a lot of work. The scale-up process and engineering from these size of bioreactors to the next level is very complicated. So I still believe that it's a simple process that can be set up locally using local EPN isolates and set up cottage industries within a country. So that is the sort of direction that we're going in. This is a 10-liter bioreactor, and we're hoping to be culturing in about 100 to 1,000 liters and obtaining similar yields. So we're well on the way to developing very small cottage industries where we can provide nematodes for local, very local communities. Cool, thanks, Steph. Now, once you have mass-produced your nematodes, you now want to formulate them and harvest them from that bioreactor. And this is what it looks like at the end of production. So on the left-hand side, you sieve the nematodes and you remove all the waste products, and you're left with what we call this paste of nematodes. And this picture in the middle, each of those is about the size of my hand, and that together is enough for a full hectare of land. So all it requires is enough nematodes that will fit into the palm of your hand, and you dilute that into how much water you need on your hectare, and that is enough to control, or that is based on our application rates, very insect dependent, of course, but that we believe is a good starting point for the number of nematodes you need on a hectare of land. What you need to do now, and this is research that we're heavily focused on right now, is formulating or developing a product that you can package and then distribute either locally or as e-NEMA and COPPERT and BASF do, then you can distribute around the world. It's a very difficult thing to keep these nematodes alive once they are formulated. So that's quite a difficult challenge that we are approaching in multiple ways, looking at different formulations to try and keep our nematodes alive after we've mass produced them. Cool, thanks, Steph.
Cool. Okay, thanks, Murray. So to translate I'm glad Murray could provide us with a very in-depth, behind the screens look at what it takes to actually get a commercial nematode out there, and translating that into work on garden weevil that both Murray and I did. So just a background, there were 3 studies over the past 12 years at Stellenbosch University focusing on lab screenings of entomopathogens, so that's both nematodes and fungi, on garden weevil. An important point to note is that no study prior to 2020 knew which weevil species to focus on. At this stage, all of the species in South Africa were still lumped under the Flectinus scolosus broader concept, but we've since had a bit of taxonomical clarification of what's going on there, and therefore it becomes important to see how different species are going to react. So larvae, pupae, and adults were screened. 9 EPN species were screened, including 8 native ones. Amongst those, Heterorhabditis bacteriophora and Heterorhabditis zealandica that Alison also mentioned, and then several Metarhizium and Beauveria native strains. So there's been very varying results, as seems to be typical with all things to do with garden weevil, but patterns do start emerging if you look over all of these studies. That is, firstly, that adults are less susceptible than larvae and pupae, and interestingly that the Steinema ergelamense, the good candidate for mass production and commercialization, gives reasonable mortality. So from what we know currently, it seems that larvae and pupae appear to be the best target for using select EPNs to try to control them. So Murray and I went and did a semi-field trial in a commercial apple block. The layout of this orchard was that the trees were planted on a raised bed. There was an interrow with a reasonable plant cover. There's a reason I highlighted that. We'll come back to that shortly. And drip irrigation. This is important because only the raised beds were wetted with this drip irrigation. So the producer who's actually on this meeting as well had to go and pre-wet the soil for us and just go and drench it very nicely. As Alison mentioned, if you've got a too low soil moisture content, the nematodes aren't going to work. So you can have a fantastic nematode, but if it's going to desiccate, it's going to die. So we raised the weevil larvae in the lab and then buried them in these little mesh cages that allows water and microorganisms like nematodes to pass through, but prevents the weevil larvae from being able to escape. These were buried 10 centimeters below the soil surface, which is about the maximum that it's estimated that most of your weevil larvae sit naturally in this interrow while they're feeding on weed roots. There were 16 treatments that received nematodes and water, or nematodes suspended in water, versus 16 control that just received water, applied in a 1x1 meter block using a watering can around this buried mesh cage. The concentration of nematodes used was 60 nematodes per centimeter squared, and the larvae in these cages was removed after 96 hours and then incubated for a further 48 hours in the lab to assess for mortality. The reason for this is that the nematodes are temperature sensitive, and although penetration of the insects and the establishment of that bacterium happened in the field, we wanted to quickly see what the actual mortality was going to be. So we can reasonably expect that a nematode that managed to penetrate the insect and establish the bacteria would have killed it in eventual course in the field as well. We just expedited that process a little bit by keeping them at the ideal temperature. So the layout of our experiment basically was that we randomly assigned treatments or controls in these 1x1 meter blocks. The insects were buried in cages 10 centimeters below the soil surface. There were buffer areas left to prevent nematodes from moving between treatments and controls. There were entire buffer rows left in order to get a bit of a bigger representative sample in the orchard environment and not just concentrated on an edge. or in a certain spot in the orchard. There were a total of 8 plots per row and a total of 4 rows. Yes, a total of 4 rows. So that comes out as a total of 32 plots, 16 control and 16 treatment. And we applied our nematodes or our water using a watering can. And for the treatments, the nematodes were suspended in the water while the controls just received an equivalent amount of water. Here's a picture of the layout. These little flags were where the mesh cage was buried. And then we wetted that 1 by 1 meter plot around that with either our water and our nematodes. And as you can see, the soil is very nice and moist due to the water drench that the producer did for us beforehand to create a nice environment for the nematodes to function. The results of this trial was that we had a control group mortality depending on the weevil species of 12 to 17% and a treatment group mortality of 22 to 45%. This 22 and this 12 is the species that is found in Australia. The control group mortality we assume is due to handling stress, but there were some other things that happened there as well. We'll get to that now. And the treatment group mortality was confirmed to be due to nematode infection. So we physically went and opened every dead weevil larvae afterwards, and you can see the nematodes inside. What's also interesting is that a specific nematode with a specific bacteria is going to color an insect a specific color. So we expected to see the little yellow-brown dead weevils for the Steinonema yergalamense that we applied, while a nematode that colors it a different color, for example red, is going to be something that's native in that soil. So we found some minor mortality due to a native nematode, which interestingly enough is Heterorhabditis bacteriophora. If that sounds familiar, that's the species that Alison also mentioned, although it's probably a different strain, and a native Metarhizium fungus. So the take home from that is that what is in your soil already is also going to help your biocontrol effort. Now, a similar study found a 70% mortality after 7 days of exposure. That was with 20 nematodes per centimeter squared, which is quite a lot less. An important point to note is that the cages that we used to bury the weevils were not as natural as ours, so there might be a bit of a discrepancy between that and our result and what's going to happen in a field setting, where the larvae are completely free moving. And it was also done on a Flectinus species that was not known at that time. So we don't know which of these two it was. However, this is quite a positive result because it seems that the longer exposure is going to lead to increased mortality over time. And given that we've got a life cycle of about 2 months where a larvae is feeding in the soil, that's a good window of opportunity to control it with nematodes or maybe even repeated applications. Okay, so the Australian perspective, on a short term, it's first to go test pathogens that are already commercially available, such as that Heterorhabditis zelandica in lab screenings. Even if it doesn't work very well, if it gives a reasonable level of control, it saves a lot of time and money to just go and register. And it says easier than it does, but it's still easier than registering or creating and registering a completely new product. But registering an existing product on a different crop and insect. So it buys you time while you are looking for more virulent or better suited alternatives. Given the fact that Alison said that Zelandica worked well in the lab, one might consider being able to just tweak the environment in which you apply it a little bit or a lot, maybe by also doing a drench of the soil beforehand with water, if that's a viable option in Australia, and also applying the nematodes at a time where the temperature is a bit higher. That bacteriophora is also an interesting option now that it's commercially available and that it operates across a wider temperature range. So that's a good nematode screen in the lab to see how virulent it actually is. Then another short-term thing is to take care of what is already in the soil. We briefly touched on that by mentioning that we found some native infection in our field study. So we've got nematodes and fungi that are already in the soil that are providing an ecosystem service. And good soil health practices like cover cropping, like that good soil cover that we had on the farm we did our field trial, and judicious use of chemicals is going to help these organisms to flourish in an orchard setting. It's important to note that they're not going to push the numbers under economic threshold, but there are still going to be a drop in the bucket towards allowing those or pushing those weevil numbers below the economic threshold, together with applying commercial products. Then much longer term is to start searching for new EPN and EPF candidates from scientific collections or new isolations from the soil environment where the weevils are present, to find candidates that might be a bit better suited to both being more pathogenic to garden weevil and being better suited to the environment. However, this is a very long-term strategy and should only be implemented if these don't already start pushing the numbers below the economic threshold. So thank you for your attention. Please feel free to contact us on these emails respectively for any questions or comments outside of the session. Thanks so much.
Thank you, Stefan. Murray, some really fascinating insights there into your current research into EPFs and EPNs, and yeah, there's some good advice for maybe some local research to go off the back of your work there. So thank you very much for sharing that. My intention was to get another speaker from South Africa, a practitioner, whether an orchardist or a viticulturalist, to talk about their sort of experiences with controlling garden weevil, or you know, their experiences with biological control agents. Unfortunately, not through lack of trying, I was unable to find an appropriate speaker, but I was able to get to have a really good discussion with a South Australian-based viticulturalist, Dricus Haynes, who works for AV&M Consultants, and being a South African himself, and managing vineyards in those regions, he was able to share some insights to me, and he noted that garden weevil is a very common pest, but like we see here in West Australia, then some seasons the pest is more of an issue than others, and also that he found it was never necessary to include an annual targeted spray program, and more of just sort of reacting when those hot spots were detected. He commented that normally he would observe the adults emerging and being noticed on the ground around the first week of October, and how important that monitoring was key to their control. One of the things that he found really beneficial in his vineyards was identifying the weeds in the vineyard that acted as host plants, and managing those plants to break the life cycle of the eggs and larvae. And he found that they were very good measures of keeping the population numbers down. The other thing he found is that making the effort of doing targeted sprays at night while the weevils were feeding was highly effective as well. Some of those points had already been mentioned in Alison's presentation as well. Now, our final speaker for today was going to be Dr. Hamish McCurdy. Hamish has just completed his PhD at Murdoch University, and now he's doing a postdoctoral research position with North Carolina State University, and yeah, he'd done in the past season. He'd been doing some work with garden weevil control in Margaret River and was going to share those insights with us. It is 4 a.m. over where Hamish is, and yeah, I'm not too sure what's happened there, but he may have slept through his alarm clock. But no judgment, because it is 4 o'clock in the morning. So if I'm able to get Hamish's presentation, I'll try and distribute that information at another time. Also, another intention to join the program was to invite a local viticulturalist who's been trialling some different approaches to managing garden weevils in WA. And again, unfortunately, I wasn't able to secure a presenter, but I was able to gain some really good insights into what's going on in the region in the space of biological control agents. And there's quite a lot of work currently being done. And based on the early stages of the development of those products and commercial sensitivity, it wasn't really suitable to discuss in today's webinar, but I'd like to just sort of bring that to your attention, that there is some product development currently underway, and some of these products could be only a few years away from being commercialized. So we'll keep our eye on those product development phases. So essentially, we're finished with our presentations. We can now open up the Q&A session. So again, just going to the top of your screen, clicking on the Q&A function there, and putting in a question, should you have one, to our presenters, and we can talk through them. I'd like to start the ball rolling with a question to Murray. Murray, in terms of EPNs, introducing EPNs into the local environment within the vineyard, is there any risk that they would start prioritizing the local beneficial insects in the soil?
That's a very good question. So there actually hasn't been a lot of work done on what you're referring to as non-targets. As far as we know, there hasn't been any reports of non-target damage on beneficial insects. I don't think a lot of people are actually doing the research to look at the non-targets. There is a bit of a, I think there's a bit of a mindset that it's safe and we shouldn't be looking at that stuff. We don't need to, which I think is very dangerous. I do think we should still be monitoring to see if something like an EPN or an EPF is targeting a non-target organism. But the way I often look at it is to say that these biologicals are the way. So chemicals, you often find with strong chemicals, you've got to bring them down in their control levels because they're so strong. Whereas with something like a nematode, we've actually got to build up its capability because they start off quite weak, and we've got to figure out how to make it better. It's not like we're starting off strong and now we're almost diluting it. So it's actually the reverse of what you see with chemicals a lot of the time. So I don't want to say that no, there are no non-target effects, because that would be a dangerous thing to say. But as far as the research is saying, if there are non-targets, it's nothing of concern. And it's at such low levels that it won't affect the ecosystem in general. So it's not going to wipe out let's say you've got a beneficial insect population that helps pollinate your plants. The EPN spraying is not going to wipe out that population at all. They never, I don't think there's ever been a case where they provide 100% control. And I don't think we never want that, because we want it to fit within an IPM program where there's multiple products being used on a single pest. So yeah, there's no danger of eliminating or literally having a micro extinction event on your beneficial insects. I don't think they aren't that powerful. They're not that good at controlling that you would get a micro extinction event. It's not like a chemical where it's wiping out everything. They can be very specific, and yeah, so there's not that much of a concern, but it's something that you do need to be aware of, I would say.
Stefan, you want to add to that?
Yeah, I just wanted to sort of tangent off that and also say, I mean, we basically just re-establishing nature in the orchard. So obviously vineyards and orchards get a bit impoverished in terms of ecosystem over time. And the main idea or the philosophy behind a lot what we do is just to go and put back what was already in the soil. So just re-establishing that natural process where there's a level of control and a level of which in this case would be a pest beneficial interaction. It's not putting necessarily an ecologically thing there that's never been there. So that's definitely something to keep in mind as well. The population of your nematode is going to spike, and then it's sort of going to go down and start oscillating as it interacts with mites and other nematodes and fungi and whatnot in the soil. So those will also stop the nematodes from becoming stop taking over the ecosystem.
Murray, you had something to add to that?
Yeah, so as Stefan was saying, a lot of the work that is now being looked at is re-establishing these populations of EPN. So when we were in France in March, I think it was March, we had a presentation from Professor Rolf Udo Ehlers, who's one of the directors at eNemo, and they said the thing that they're noticing is that it's not often that you have to apply EPNs for a specific insect pest. They're actually trying to rebuild populations that have been diminished because of mass chemical spraying. There was actually quite a scary study that was released recently showing the effect of glyphosate or Roundup on beneficial nematodes in the soil. So in an untouched and or maybe even an organic setting, there would be a strong population of EPNs existing on your soil, acting as a natural buffer against any kind of insect pest invasion. But with mass chemical spraying, we've actually destroyed a lot of those EPN ecosystem communities. And now what Rolf or Prof. Udo Ehlers is saying is we actually need to rebuild those communities by spraying these EPNs. Now, what we say is what you should do is go to an orchard, assess what their EPN, the level of EPN, the size of the EPN population in that soil, take that EPN and mass produce it, and then put it back into the soil to the levels that it was before you started spraying chemicals, or at least something similar to that. So a lot of the work that we're trying to now direct ourselves towards is re-establishing EPN communities in these agricultural soils because they've been so badly diminished by previous agricultural practices, particularly chemical spraying. So that, yeah, as Stefan says, there's a bit of a re-establishment aspect to EPNs, not just using it as a product to control insects.
Thanks, Murray. Yeah, I think it's probably in most viticulturists' aim is to have that balanced ecology. Obviously difficult to know what the starting point was before conventional farming occurred on the land there, but yeah, definitely in the mindset of local producers here in WA. And yeah, thanks for your answer. Alison, Stefan's put a question to you about the detail on the bait that was used for garden weevil.
Yeah, so in that project, they were looking at what's the possibility of using a bait that's, I guess, spread onto the orchard floor. So you're not actually putting any chemical into your trees, onto your crops. And the first step in that was coming up with a bait that's attractive for garden weevils to eat. So they tried, it was all just done in the lab, and initially they tried a whole bunch of, I guess, food products that they thought they would work, and the ones that worked fairly well were carrot and apple, and then they did like a prototype pellet of pelletized form of that, and they still got I guess what they thought was fairly reasonable attractiveness of the weevils to those pellets. And then, yeah, it said in the report, the next step is then obviously, putting a toxicant in that and trialling that. But as far as I'm aware, that next step, it didn't happen in that project, and I don't think it has happened as far as I'm aware.
Thanks, Alison. Thanks, that's fascinating.
And another question to you, Stefan, if I may. If you had to back a horse in the EPN or the EPF race, which one of the two do you think is more effective? And is it not that simple? Is it a combined effort that would yield the greatest results?
That is a difficult question to answer. I am slightly biased towards EPNs.
That's why I didn't ask Murray, because he's 100% biased.
There's a couple of reasons for that, and I'm going to say why I am biased as well. Firstly, in order to get an EPF to larvae and pupae in a perennial system with garden weevil is going to be, in my opinion, not worth the cost. You're going to have to do soil drenches, and since fungi are dosage dependent, it depends on how many Canidia are going to physically adhere to that insect. Given that you've got an insect that's feeding inside the crown or inside the root of weeds or your crop, to actually get that fungus, and obviously it doesn't cost negligible amounts to apply it or to buy it either, to get it onto that target is quite difficult. So that only leaves adults as a viable life stage to control with EPF. Now, the adults with a very few fungi that I've worked with weren't very susceptible, and in my trials the conditions were biased to suit the fungus and not the weevil. In the field, it's probably going to be a different way around. Fungi don't do very well in exposed environments like foliage. The UV tends to fry them quite quickly. So in order to once again get enough spores onto an adult is going to be difficult. Now, there are maybe ways to do this. For example, if you go and put a targeted spray around your trunk or around your crown or the butt of the vine or the tree that the weevil then needs to walk through, that might be a viable option. Now, the nematodes, the reason I'm biased towards using them is that they seem to work rather well on the larvae and the pupae, and they're also going to kill a few adults while they're at it. If you put it on your soil bed and on the weeds and everything that's growing, a cover crop that's growing there. They can physically move and they can go find the larvae and the pupae. Murray always describes them as these little wolves because they go and hunt and they go and find the target insect. But it is probably going to be a combination of strategies that will be most effective. And we are hopefully going to conduct some more research on entomopathogenic fungi next year, and we'll hopefully be able to give a bit of a better answer on how we can overcome the challenges associated with using fungi in the field.
Okay, thank you for that unbiased answer, Stefan.
Pleasure.
I appreciate that. Doesn't look like there's any more questions to come from the audience. So therefore, I'd like to wrap up today's webinar by thanking our panelist speakers for their time sharing their knowledge and experience with garden weevils, EPNs and EPFs, an acronym that I'll try and recall. And I would like to also acknowledge Wine Australia for funding this event through their regional program, which is administered by Wines of WA. So yes, good luck with your garden weevil management. Very exciting to know that there's product development underway, and I look forward to keeping you posted on how those developments pan out in the coming years. So thank you and goodbye.
Thanks.
Thank you. See you.